In this lecture, we will learn about the understanding the design concepts of bioimplants. In the field of bioimplants, if any student or a researcher is entering, they basically are worried about the applicability part of the bioimplant. So that is the reason it becomes very essential to learn a few design concepts, or to learn what are the parameters or what are the conditions which will decide the applications of bioimplants once it is inserted into the body. So the applications are the very essential part of it, because that decides the overall usability of a particular device material. So there are certain so synthetic biomaterials, they basically go into applications such as dental applications, sutures for hernia repairs. They can also be utilized in vascular grafts or stents, drug delivery, and non-adhesive soft membranes. So you can see a myriad applicabilities of synthetic biomaterials. Those are not natural biomaterials but synthetic. So that is the reason they have to basically undergo certain selection criteria, such as for dental applications, those are again more load bearing. They need to also have fracture toughness, and again for the sutures, they need to dissolve with time, or they have to be removed if these are non-dissolvable or non-degradable polymers. So again there can be vascular grafts, there can be stents for basically doing the repair of broken veins or arteries in a heart, and it can again be for drug delivery, for releasing certain drugs at certain required area, and it could be again non-adhesive soft membranes to avoid the sticking of two nearby tissues. So these are certain applications which are very essential for utilizing synthetic biomaterials. So it becomes very essential to understand these applications, to understand what are the parameters which will decide the applicability. So the applications have been there for since last 40-50 years. So with their usage, there have been certain failures as well as certain successes. So the degree of success and failure also decides the longevity of a particular device which is implanted into the body. So the degree of success and failure will tell us what we can learn from the evaluation of implant history. If the implant has survived, then what was the reason for it? Even if the implant has failed, what was the reason for it? So we can always improvise on what the devices have been utilized for. Again, there are certain criteria which becomes very critical, such as blood contact. So if a device is coming in contact with blood, there might be thrombosis or certain inflammation which can release into the nearby adjacent tissues. It can also be drug delivery for the controlled release of certain drugs into certain areas. It can also be utilized as a sensor for diagnostic purposes. In certain situations, it becomes essential that we can extract some data or get some diagnostic from a particular blood sample. So we can also have certain sensors which can exactly sense. It can be the glucose level, it can be the drug release, or it can be any chemical composition of a particular area. So that is again required for the sensor application, and again extending and improving the current application. That again comes from the understanding of the failure or the success of the implant from its implant history. So that becomes very essential. So again we need to also extend and improve the current applications. So only once we have learnt enough, then we can apply further. For at the same time we should be able to improvise on what we have learned, and then develop or extend the applicability of a certain device. So that is the reason we concentrate much on the extending and improving the current application. So certain applications which were not possible earlier can again be thought of. And again those applicability again it has not gone up to the expectations. The general implant life is approximately 15 to 20 years, but in order to sustain that particular implant for a much longer time, we need longevity which is 4 times as that of a current implant materials. So the longevity or the time period of an implant in the body we are looking is around 80 years. That is approximately 4 times improvement, that is what we require from the current used implants. So just reiterating that the understanding the applications is basically based on the degree of success and failure, and that we can learn from the history of that particular implant. And that particular implant can have some sort of a blood contact or tissue contact. It can again be utilized for drug delivery. So for controlling the particular aspect of the degradation part, we again require certain engineering. Again for sensing, so sensing also has to be very peculiar for a particular place, and for what purpose we are utilizing it. It can be for diagnostic purpose. So, we should be able to evaluate the chemical composition or apply certain drugs to a certain area and so on. So, again we should not limit our applications to what we have been utilizing, not only for improving the material, but also improving the applicability. So, we have to extend the limits of current device materials and be able to extend it for certain other applications as well. And also we require longevity of those implants, because if an implant is inserted, it requires surgery. So, to remove the surgical complications, or to remove that costly surgery which is done again and again, maybe a couple of times for a particular patient which has to undergo surgery, it becomes necessary that surgery time can be reduced, or the number of surgeries can also be reduced by extending the lifetime of those implants. So, in this we can concentrate on the basically on the cardiovascular devices. So, in the cardiovascular devices, we can see that if device is implanted, it has to interact with the blood as well as with the surrounding tissues. So, it can happen that there can be addition and growth of certain bacteria or infectious agent near the device. So, that basically will hamper the overall applicability of this particular device. Later on, there can be formation of thrombi on the device surface, that will again limit, because that now the surface of the device is now covered with certain fibrous thrombi. So, that can basically limit the overall applicability, and further this device can also lead to injury of the cells by its basic functionality of the tissues. So, that can also hamper the overall applicability of the device. Also, it can cause injury to circulating blood cells. So, again that may also need to be considered while a particular device is being implanted. And again there can be intimal hyperplasia. So, there is excessive growth of cells, which can again result in the reduced flow, because if a particular lineage is there, then it can have certain hyperplasia. So, excessive growth of cell, and that can again reduce the overall flow because of the reduced area which is available for the blood to pass through. So, these are the critical issues with the cardiovascular devices, because it retains in contact with blood and with the surrounding tissues. So, the addition of it can lead to infection by the growth of bacteria or some infectious agent into the device, and it can again form the thrombi on the device surface, that will again limit the overall exposure of this device with the surrounding. And again it can cause certain injury to cells or tissues near the device, and it can also injure the flowing blood cells, or it may also lead to hyperplasia, which can result in the reduced flow of a bloodstream. So, it becomes very essential to evaluate the blood material interaction, because as soon as a particular material or a foreign material is implanted into the body, there is an immediate adsorption of proteins or lipid layer on the surface. So, as soon as we have a device, it basically gets adsorbed by proteins or lipids, and it is followed by the addition of the platelets, leukocytes or erythrocytes, and later on there can be formation of pseudointima on the inner surface, and that basically leads to the formation of tissue capsule on the outside surface of the device. And once this process has taken place, it can again change the mechanical or functional properties of the device. It can be both beneficial, or it might be deleterious, but this is what is happening in the bloodstream after the material has been implanted, which is in contact with the blood. So, we have the device. So, as soon as we insert the device, we see absorption of proteins or lipids on the surface of the device material. It is followed by the addition of platelets, leukocytes or erythrocytes. So, basically the blood protein they get basically adsorbed on the surface, and the formation of pseudointima, which results on the inner surface, and that results the formation of tissue capsules. So, if you have a particular device, it will have first the adsorption of the adsorption of proteins or lipid on its surface. It is followed by the addition of platelets. So, we have platelet addition on the surface, and then this formation of pseudointima over this, that forms on the inner layer of the cell or the tissue, and then it is on the outer surface. This is the device, and this is nothing but the tissue. So, the in between layer basically gets the pseudointima formed between the on the outer surface of the device or on the inner surface of the tissue or the capsule. So, that encapsulates the device, and that once the device has been encapsulated, it might be beneficial in terms when we require some sort of a cell adhesion on the surface, or it may not be really required, it might be dull interaction, and that basically affects the mechanical or the functional properties of the device. Again, there is second application, which is cardiopulmonary bypass. It is also called as the heart-lung machine, or extra corporeal circulation. That is basically required because we want to exchange the oxygen when the heart has been disabled. So that basically results from replacing or improving the functionality of the heart. So in this particular case, the cardiopulmonary bypass, it helps in pumping the unoxygenated venous blood. So we have the venous blood which is full of carbon, which does not have enough oxygen. So oxygen has to be reactivated. So the oxygenation of this particular venous blood has to be carried out using certain oxygenator, and that can be done via certain oxygenator, which can be either bubble or membrane type device. So basically, what is happening here is the cardiopulmonary bypass, it pumps the unoxygenated venous blood from the right side of the heart through certain oxygenators instead of the lungs, because this particular blood has to get oxygenated via lungs, but now this synthetic oxygenator can take care of the oxygen exchange in the venous blood. And this particular development has led to easiness in terms of doing heart surgery, because heart has the blood which is flowing through the heart has to be oxygenated. It has to have sufficient oxygen to supply to the all the regions of the body. So instead of getting the oxygen from the lungs, it can allow oxygen exchange using this particular synthetic oxygenator. So this particular device, which is cardiopulmonary bypass, this synthetic oxygenator, it basically includes gas exchange device, which can be bubble type or it can be membrane type. In bubble type, the oxygen bubbles are basically being released, and then later on it requires defoamers. So because when bubbles are created, it starts forming certain foams. And this membrane type, it basically filters out and then it releases the oxygen. So this is much more nicer way for the blood to trap the oxygen, but the process is very complex and it is very costly as well. So these both practices are being adopted. Either it can be membrane type or the bubble type to exchange the oxygen flow, and it also has certain blood pumps, heat exchangers, ventricular vents, or pericardial suction line for certain operations. It also has tubings, filters, and reservoirs for carrying out this particular bypass. So in this particular case, we can see cardiopulmonary bypass. It basically is also called heart-lung machine because it supplies oxygen. Instead of letting the oxygen come from the lung, it supplies the oxygen. It is also called as extracorporeal circulation. So in this particular case, it will pump out the unoxygenated venous blood from the right side of the heart through certain synthetic oxygenator. So basically, it has enabled the heart surgery, and the synthetic oxygenator includes gas exchange device, which can be either bubble type or membrane type. In bubble type, there is a release of oxygen bubbles, which basically go and react with the venous blood to exchange oxygen, or it can be again membrane type. But the bubble type starts creating certain froth, so it needs a defoamer, whereas membrane type is much more softer in operation, but it is very complicated as well. It requires much complicated devices to take care of this particular exchange of oxygen and passing of this particular blood. And it requires certain other devices such as blood pumps, heat exchanger, ventricular vent, filters and reservoirs and so on. So in order to carry out this cardiopulmonary bypass. So we can see that how complicated this particular process is in terms of utilizing a bypass. Further, there are certain other devices such as heart valve. So first of all, this prosthetic heart valve was first utilized in 1960 in humans, but again it basically had certain problems, and it basically had certain moving component which are called occluder. They can be made up of either cage ball or tilting disc. So in this particular case, we have a sort of cavity, and in that we have certain fins or tilting discs. So these are sort of a disc which will basically alter depending on the flow of the blood through them. So that is what is basically there, and they respond passively to the changes in the pressure of flow, and they are made up either of titanium, cobalt, chromium, silicon, or pyrolytic carbon. So these particular heart valves are utilized. These are nothing but the valves which will have to turn or tilt. They can be either ball type or tilting disc type to alter the or to direct the flow of the blood, and they can respond very passively to the changes in the pressure of flow, because once the pressure is high or low, they have to alter the overall flow of the blood through the valve. So valve is nothing but controlling the flow of the blood when the pressure is high or low. So again they have to respond accordingly to that. So they have certain either fins or balls which can, and in this particular case they have some sort of a disc which can leaflets which can alter the directionality of the flow. Again there can be these are again synthetic walls or prosthetic heart walls. There can be either bioprosthetic or tissue walls, and this can again be prepared either from homografts or allografts which are prepared from the human body, or they can be heterografts or xenografts which arrive from the foreign body or prepared from the tissues of another species. So again there are two sides of the heart wall. They can be the synthetic, and it requires some sort of occluder which is nothing but a moving component. So we have certain disc available, and then it has a caged ball or some sort of a disc which can allow the directionality of or change in the pressure or flow, and they are made up of titanium, cobalt, chromium, silicon or pyrolytic carbon. At the same time, they did not get they need to control the flow, so they need not really get damaged by the flow of blood itself. And these can again be bioprosthetic or the tissue walls, and these are more natural. So they can again come out either from they can be prepared from the human body, which are called allografts, or they can be prepared from the foreign tissue of another species. Those are called xenografts. Again, these vascular grafts they can be either biological or synthetic. So first coming to the venous autografts, auto means it is from the self part of the vein. So it comes from the patient's own calf vein, and again the autologous saphenous vein, which comes out from the calf of the calf vein of the patient, it basically performs much more satisfactorily. But again it can induce certain problems, like 30 to 40% people can face problem in case when there is no suitable saphenous vein, or there can be small or phlebitis inflammation if inflammation has been occurring, or there is or that particular vein has already been removed. So there can be certain problems which are associated with the venous autografts, that there is no saphenous vein available inside the body, or that particular vein has already been infected, or it has already been removed by some previous surgery. But they show best result in the reconstructive arterial surgery, which is below the knee. So they are well suited for the arterial surgery which is below the knee. So they have shown very promising results. They have also been successfully used for either coronary, visceral, renal or peripheral arteries, both for replacement as well as the bypass surgery. But the basic problem comes from the vascular grafts is it can fail by thrombosis, which is followed by neo-intimal hyperplasia. It means there is some blood clotting which can be follow by the newly which can form after a new surgery has been done, and there is a formation of new cells which basically which is basically excessive in the in a normal growing tissue. So that problem can occur in the vascular graft, that there can be excessive formation of cells in an abnormal way in a normally growing tissue. So again we can see the vascular grafts, they can be biological or synthetic. Venous autograft, they come out from the patient's own calf veins. So that is the overall autograft, the body will start reacting to it in a more friendlier manner, because that particular vein has come from the body itself, and they have performed very satisfactorily. But the problem comes when that particular vein is not available, or it is inflamed, or it has already been removed by the surgery, and they show very good results when in the arterial surgery of the knee. And they have also been utilized for coronary, visceral, renal and peripheral arteries. But the problem comes when there is occurring of thrombosis, which is followed by neo-intimal hyperplasia, or the excessive formation of cells in a normally growing place after surgery has been taken place. There can be other venous homografts. So again the homografts which are taken from the venous parts, and they were discontinued in 1950 because of their high failure rate, and it happens because they are antigenic and they sensitize the recipient. So that is the reason they were discontinued in 1970s. Further, there can be arterial xenografts. So these are taken from the arterial regions from the foreign body, and they were widely used in 1950s. But again the higher rate of thrombosis was observed, and higher rate of rupture was also observed in this particular case, and they were discontinued later on again in 1970s. And it also was seen that they can also create infection. What happens in the arterial xenografts is they develop inner lining of host derived fibroblast, So that is the problem that occurs, that either it can lead to a higher rate of thrombosis, and it has also seen that the rupture rate was very high, and also it led to the infection later on. The problem with them is they develop inner lining of host derived fibroblast, but they have seen to perform better in the aortic and the iliac positions. So these arterial xenografts are well suited for aortic and the iliac positions of the heart. There can as well be, apart from the autograft, there can be synthetic grafts as well. So for the synthetic grafts, they become a requirement, because sometimes in autograft it may not be possible that we have a particular vein available, like in aorta, which is a very large vessel, So in that particular case, it may not be possible to extract a different aorta sized vein from the body, or aorta sized vessel from the body. So it becomes a requirement that we produce synthetic grafts. In certain cases, even thinner vein vessels may not be also available. So in that case also, we need to extract, or we need to make or synthesize a artificial graft for the body. Again, they are required for very large vessels such as aorta, or even for very smaller ones. This performance becomes unsatisfactory when the performance of the homograft or the xenograft becomes unsatisfactory. Then also we need to make the synthetic grafts, or if there is any unavailability of the suitable autologous vein for either coronary or small artery grafting. So either when we require very large vessels, or when we require very small arteries, or when the performance of the homo or xenograft itself is unsatisfactory, then we need to go for the synthetic grafts. In the synthetic grafts, the healing occurs by the formation of fibrous capsule around the graft, and then there is a formation of pseudointima on the luminal or the inner surface of the cell, and then there is a lining of fibrin which gets covered by the fibroblast or macrophages, and then healing is aided by porosity, which allows cell ingrowth and also allows the nurturing of the pseudointima by the capillary which are present in the pores. So we can see in the synthetic grafts, it becomes a requirement for large vessels such as aorta, or even for the small artery grafting. That actually occurs when there is unavailability of suitable autologous vein, or when the performance of the homograft or the xenografts is not up to the mark. So in that particular cases, the synthetic graft becomes a requirement. In the synthetic grafts, the healing occurs by the formation of certain fibrous capsule around the graft. So if we have a graft, we have some sort of fibrous capsule around it, so it encapsulates it. Then formation of pseudointima. So we have a cell surface. So in the interfacial region of the graft and that of a cell, in the luminal surface of that particular tissue. So we have tissue, and then the pseudointima forms on the luminal interface, and then we get a lining of fibrin. So we get a lining of fibrin over it, which is now covered with the fibroblast or macrophages. And in this particular case, if we have certain porosity, so we have certain porosity which is available, then what can happen? Then it can also lead to the nurturing of the pseudointima. So the pseudointima which is formed on the surface of this particular device or this synthetic graft, it allows nurturing of that pseudointima, and it cannot form natural intima. So it needs to have a pseudointima over there. So it can allow the nurturing of the pseudointima by the capillaries which are present as in between the pores. So if you have certain porous region, we can allow the porosities, or the porosities can allow capillarity, which can allow formation of this capillarity, and then that can nurture the pseudointima. So we can see the healing is occurring by formation of fibrous capsule around the graft, then formation of pseudointima on the luminal surface, then there is some lining of fibrin which gets covered by fibroblasts and macrophages, and then healing is basically being aided by the porosity, which allows cell ingrowth by nurturing through the capillaries. There is another device which is called stent, and this stent is is basically nothing but a scaffold to maintain or increase the lumen of a vessel. So if you have a vessel, so if you have a particular vessel, then we need the lumen of it. So this is nothing but the internal lumen of a vessel, the inner lining of the vessel, that there is what is the overall diameter of that particular vessel. So it has to maintain or increase that particular lumen. So in many cases there is some deposition of certain proteins, or some sort of a decrease in the overall lumen of a particular vessel. Then it becomes very hard for the blood to flow through it. So, in that particular case we require stents to allow the flow of blood, which is basically unperturbed. So, in this particular case, if the vessel itself is basically constricted, so we need to send some artificial material which can again lead to the expansion of this particular vessel and allow easy blood flow through it. So, it counteracts restenosis by the percutaneous transluminal coronary angioplasty, which is called nothing but balloon angioplasty. So, in certain cases, once we have done a surgery and then basically we see the retinal or the lumen of a particular vessel starts decreasing. So, that again reduces the blood flow. So, we need to counteract that by again increasing or ballooning this particular vessel for the smooth blood flow. So, there are certain designs which are based on this which are available for the stents. First thing is spring like design which expands. So, we have some sort of a spring like design which can expand itself once it is going through it. So, initially we can have a compressed spring which upon going inside, the overall restraint to it can be released, and then basically they grow and again increase the diameter of this particular vessel. So, initially when we have smaller vessel and then the spring goes and start expanding to further increase the overall lumen of a vessel. There can be also thermal memory stents. So, in this case this material nickel titanium, this is called nitinol. This is a shape memory stent. So, it regains its shape once it is exposed to the temperature of the blood. So, nitinol is a material which is inserted into the vessel, and then it starts to increase its shape. Thus, it can allow the pipe or the tubing to enhance in diameter. So, that's what is happening once we have this particular stent, that we take a particular vessel, we insert the stent part into it. So, we have some sort of a tubing, and then basically it is inserted into the body, and as soon as it experiences the body temperature, which is nothing but 37 degrees centigrade, this ballooning expands. So, this particular ballooning particular expands, and then it increases the diameter of the blood vessel. Again so these are certain designs which are spring like design, thermal memory stent, or it can be balloon expandable. So, in balloon expandable we have a particular tubing, we insert certain balloons and we have a stent over it, and once we start ballooning it, once we start filling in the air in that particular balloon, we have also a stent which is lying over the balloon. So, that particular stent also grows in size along with the balloon. This is nothing but the balloon cavity which basically starts filling in with air. So, the overall lining around it, that is nothing but the tubing or the lumen of a vessel, it also starts increasing. So, we can see that balloon expandable stent can also increase the overall diameter of or the lumen of a particular vessel. There can be fourth type, which is stent of a biodegradable polymer. So, we can again have certain biodegradable polymer which can be inserted as a stent, and then it can also start releasing certain drugs which are required for the along that particular regime. So, we can have certain cardio drugs which are available for reducing the thrombosis out there. So, we can see there are certain designs. Overall, there are certain stents which are required for maintaining or increasing the lumen of a particular vessel, and it counteracts the restenosis or the reduced blood flow by percutaneous transluminal coronary angioplasty, or which is also called balloon angioplasty. It has certain designs which can be either spring like, which can expand, it can be thermal memory stent which can again retain its original shape. So, basically we take a stent, we compress it, and then we insert it into the tubing, and upon experiencing the temperature of the blood, it again starts regaining its original shape, which was much bigger diameter, and in process it increases the lumen of a vessel. And it can also be balloon expandable stent. In that particular case, we have a particular balloon which has a stent over it, and as soon as we pass the gas or air into that particular balloon, it increases in diameter, and in process it also increases the diameter of the stent, which remains there for a longer time, and in the process it has increased the lumen of the vessel. And it can also have, the stent can also be type of a biodegradable polymer, in which case we have a degradable polymer which goes in and starts dissolving so that the original lumen can be restored, and with time that polymer starts dissolving and provides the space for the blood flow. And again, this biodegradable polymer can also carry certain drugs if required for the cardiovascular surgery. And there is another class which is called catheters. These are nothing but intravascular tubings which are placed either in the arterial or the venous circulation. So, these catheters are the tubings which are basically required for either administering fluids. So, in certain cases when a surgery is done, we need to introduce either blood products, glucose or medication, then these catheters come to our rescue. They are also used for obtaining certain data such as what is the pressure or the blood samples for certain chemical analysis. And so that is the overall applicability, that catheters are intravascular tubings which are either placed either as in the arterial or the venous circulation, used for administering fluids which can be blood products, glucose or even the medication. They can also be utilized for the obtaining the data which can be the pressure or blood samples. But the problem with catheters is that it can lead to thrombosis. Again, one more requirement of a catheter is that it requires much more flexibility, and also its chemical composition should be suitable so that it should not become an issue later on. And there are certain materials such as polyurethane and silicon which are utilized successfully as catheter materials. So, again the catheters, these are nothing but tubings which require to either administer fluids or gather some data which can be pressure or blood. But there are certain issues which can come as flexibility of the chemical composition of the catheters, and that might lead to thrombosis for long term usage. So, again certain materials such as polyurethane or silicone have been utilized as catheters. There is another class of device which is called pacemaker, and this is utilized for rectifying the abnormal heart rhythms. So, what a pacemaker does is it generates a pulse which has certain lead connectors, electrodes and certain lead wires. So, basically we have a pulse generator which is hermetically sealed, battery which is enclosed again in a titanium, and then electronic module will communicate through external connectors, and these external lead connectors permit continuous communication between electronics and the pacing lead. So, what has happened there is it can also the lead connectors or the electrode tip should also have certain steroids which will increase the simulated threshold, which are there to restrict the simulated threshold, because in certain cases that can occur by the cell attachment or the growth on the tip. So, if you have a particular electrode, it can have a certain cell attachment or the growth on the tip. So, we need certain steroids to prevent that increase in the threshold. So, that particular part is restricted by inducing certain steroids on the tip of the electrodes. So, we can see the pacemakers are utilized for rectifying the abnormal heart rhythms. It needs to have a particular heart rhythm to keep the heart beating. So, it generates a pulse in a particular frequency which are connected with lead connectors, electrodes and wire, and that particular pulse generator is sealed. It is sealed and which is encased in the titanium alloy. Again, the electronic module will communicate through external connector which basically permits continuous communication between the electronics and the pacing lead while providing certain fluidic sealing to it. So, that is what is there, and then again there are certain steroids which can be induced on the tip of the electrode to prevent increase in the simulated threshold. If it goes beyond that, basically there can be excessive beating of this particular heart, and it will lead to increased pressure in the system. Again, then one more problem can be eliminated by utilizing total artificial hearts. In this particular case, what happens is this total artificial heart replaces the mechanical functions of the heart, and this is required when heart fails irreversibly, and again when heart transplant is not possible. So, but this total artificial heart can induce some thrombogenic complications, lead to infections and failure of other organs such as kidneys or liver. So, in that particular case we require total artificial heart surgery. But again this total artificial heart surgery is very much essential when we do not have a donor which is available right now for a heart transplant. So, ideally since they cause much more complications, infections and failure of other organs, they are basically utilized for some shorter terms from days to weeks. Also there is some other device called ventricular assist devices. Those are utilized until the donor becomes available. So now we are utilizing either TAH or VAH for shorter duration until we have a next donor available to donate a heart, and this becomes a requirement when heart has completely failed and when the heart transplant is not immediately possible. So instead of their longevity, these are being utilized for short duration from days to months or to weeks. Again, then ventricular assist systems, they come in blood contact. So we have certain in the ventricular assist systems, we have many materials which becomes of importance, such as for the blood contact. For the blood contact, we have polyether or polyurethane, and the blood contacts regularly happens either in pumps or flow conduits or valves. So in that particular case, when the blood contact is occurring, we need to utilize certain materials which can be polyether, polyurethane to assist that total heart replacement. It can again be tissue contact. So again, tissue contact will require certain materials such as titanium, which is commercially pure, silicone, epoxy, polypropylene or polyether. Again, these are basically come when we have a belt skin transformer which has certain interconnecting leads, certain drive units or even conduits. So this tissue contact will becomes of prime importance when the device itself is coming in contact with the tissue. So in that particular case, we require materials such as titanium, silicone, epoxy, polyether or polypropylene. Again, there can be certain special structural materials which need to bear the load or need to perform certain functionality. Those can be solenoids for generating the pulse. It can be volume compensator, again belt skin transformer, can be gas filled reservoirs or even batteries. And these materials require materials which can be titanium, vanadium, copper coils for the solenoids, nickel, cadmium, even silver or copper for connectors. So we can see that overall, either for total heart replacement or the ventricular assist system, we require the device to be in contact with the. So we can see that either in total heart replacement or the ventricular assist systems, blood contact and tissue contact becomes of prime importance. So in certain such as pumps which are flow conduits or even valves. So there we need to see, we need to particularly design a particular device so that it can take care of itself, and we need to have certain specification for certain materials which can come out to be polyether, polyurethane, silicone, epoxy or titanium. Again, there are certain structural materials which can be also required for performing the structural or the weight bearing part of it or any other functionality which can come out from the solenoids, volume compensator, belt skins, gas filled reservoirs, batteries. So we require certain materials which can be copper coils, nickel, cadmium, silver, vanadium, titanium. So we can see the overall functionality of this particular device depends on the overall functionality of a particular material which can be utilized in certain regime. They can again be blood substitutes. Again, blood substitution depends on the healthy and a willing donor. So that is very much essential that the donor has to be willing and as well as healthy. But again, the blood transfusion can create certain problems which become a mean of transmitting diseases, which can be even hepatitis or even AIDS. Blood basically contains certain separable therapeutic components which can be either erythrocytes, leukocytes, platelets, plasma proteins, albumins, fibrinogen and even other coagulating factors. So we can see that blood basically has so many constituents, therapeutic components which can be separated, but blood substitutes refers to the oxygen carriers which basically is utilized for substituting erythrocytes or red blood cells. So the blood substitute is nothing but it refers to some oxygen carriers which can contain the oxygen and then it can substitute red blood cells. Again, it has the blood substitution has to depend on a particular or willing or a healthy donor, but blood transfusion can create certain problems. It can transmit diseases such as hepatitis or AIDS. So apart from so many constituents, we are mainly concerned about the erythrocytes or the red blood cells to take the functionality of the erythrocytes in terms of oxygen scavenging. Again, there are certain materials which is something called perfluorocarbon which can carry as much oxygen as hemoglobin. Again, they can be alternatively applied by encapsulating erythrocytes by cross-linking hemoglobin and basically separating it from the stroma. But the problem with encapsulation is that it has a very short survival time when it comes to the circulation, and it is immediately consumed by the reticuloendothelial system. So, once we have the cross link or encapsulated hemoglobin, once after it has been separated from the stroma, its lifetime in the circulation, it's very short, because it gets consumed by the reticuloendothelial and endothelial system. So, the overall basically aim in cross linking this particular hemoglobin is that it can prolong its survival in the circulation. So, once we have hemoglobin and we cross link it with certain polymer, then it can prolong its survival in the circulation, and that becomes very effective once we are treating some acute hemorrhagic shock. So, once there is much more problem in treating a particular hemorrhagic shock, then we can supply hemoglobin, which is again cross linked, and then it can reduce the damage by cross linking it to the polymer and then releasing it into the circulation stream. So, in certain hemorrhage cases, it can be effectively applied once it is cross linked to a certain polymer, because that will prolong its survival or the overall survival time in the circulation. So, that can basically assist in recovery. So, we can see again blood substitutes. Perfluorocarbons can carry as much oxygen as hemoglobin, so they can be effectively utilized. Alternatively, we can encapsulate this hemoglobin or cross link the hemoglobin, and then once we have cross linked it, then it can the survival in the circulation becomes much more prolonged, but encapsulation will make it get consumed very quickly by the reticuloendothelial system, and this becomes very essential. The cross linking becomes very essential once we have certain acute hemorrhagic shock. Again, the future directions basically go on to evaluating the long term exposure. So, in this particular case, we have to see the long term exposure of devices to bloods or tissue. So, because the short term is very easy, and it can basically suppress the overall infections, or it takes certain time for the cells to respond to a particular material. So, short term exposures of devices, they are now very much limited, and the future direction is phased towards long term exposure of devices to bloods and tissues. So, the device has to perform satisfactorily under the contact of blood or tissue for a prolonged time. So, that is where the future is basically going. And again, interfacial events, they become of prime importance, because as soon as this particular device is inserted into the body, it will observe certain protein adsorption, then release of certain harmful bacteria or even infection, and again there is some formation of fibrin or fibrinogen or macrophages over the surface, formation of pseudo-intimal tissues or such features. So, that basically can degrade the overall functionality or the mechanical properties of the device. So, it becomes very essential to study the interfacial events which occur after a long term implantation. So, again long term exposure of devices to bloods and tissues, and seeing what is happening at the interface between the device and the surrounding tissues after long term implantation. Such as something has been proposed for the cardiovascular devices, that it needs to have an inert implants, because we do not want any tissues to grow on it, otherwise it will start reducing the lumen of a particular vessel, or it will reduce the functionality in terms of either pacemakers which need to be free from any cells, or even drug delivery which needs to supply the drug, or even sensors, because sensors need to be exposed to the bloodstream continuously. So, overall inert implants can be inert implants has to be utilized for the cardiovascular devices. And again, the overall functionality which is required by the cardiovascular devices is that it should release molecules which are similar to the organ which it is replacing. So, the overall implant has to release molecules which are basically similar, so that the overall functionality of the organ can be mimicked by the device, and again they should become integrated with its biological environment. So, if a particular device has been now inserted into the body, it should perform as it is natural to the body. So, it should become integrated with the surrounding tissues or the biological environment, and also it should provide mechanical or functional properties of the organ. It should sustain the mechanical loads which a particular organ has to sustain, also perform the functionality of a particular organ which it is replacing. So, that is where the overall natural future direction is leading us to. It is long term exposure of devices to bloods and tissues, considering the interfacial events even after long term implantation, and the usage of inert implants, so that there is no toxic or any adverse effect which arises from the surrounding cells or tissues, and they should release molecules which are similar to that of a organ which it is replacing. It should get integrated with the biological environment, and it should also provide mechanical as well as functional replacement for the organ which it basically is going to serve. So, in summary, we did see that the overall blood material contact is becomes of prime importance, that as soon as blood is basically comes in contact with the material, it starts depositing adsorbing proteins on the surface, then releases certain actin agents which starts forming the fibrins or the fibrinogen or the macrophages, and then forms a pseudo-intimal lining over the implant. So, that can be either helpful in certain cases, maybe deleterious in certain cases. And then we also saw that heart valves, they can be either of the ball type or the tilt type. So, that can be utilized for assisting the blood flow or catering to the pressure changes in the valves. So, these valves have to tailor accordingly. Again, there can be vascular grafts. These can be either autografts or the xenografts, depending on where the grafts have been taken from. Stents, these are again utilized for improving the lumen of a particular vessel. They can arise again by ballooning or by spring like. Those can again be from the shape memory nitinol type, or those can again be rising from the biodegradable polymer. So, again stents they are utilized in terms of restoring the lumen of a particular damaged artery. They can be again catheters for excavating certain data or information, or even supplying nutrients to a particular regime. And there can be again pacemakers for restoring the rhythm of the heart. So, that in that particular case we require pacemakers for actuating the rhythm at certain frequency. There can be again total artificial heart for in certain cases when we do not have a donor rightly available, or when the total functionality of the heart is being hampered. So, for certain duration, for short duration from weeks to months or even from days, we have to wait until the donor is ready to donate a heart. So, for that particular time we need that the functionality of the heart has to be totally given up to certain machine. And there can again be blood substitutes basically for the erythrocytes or red blood cells, when the oxygen scavenging has to be being taken care by certain materials. So, we did see that we can either encapsulate it, or we can supply with a material which can store the hemoglobin for certain duration, but by storing the hemoglobin or encapsulating it, it gets consumed very quickly in the circulation. So, we need to supply certain cross linking to polymers which will prolong its survival in the bloodstream. So, we can see the overall applicability of this particular devices becomes of major importance, because of it undergoes blood material contact, utilized in heart valves, vascular grafts, even stents, catheters, pacemakers, even for total artificial hearts and blood substitutes. But the overall functionality here is very, very different. In some cases we require certain cells to form certain pseudo lining to form, pseudo-intimal lining to form over it. In certain cases we do not require any cells to form on it, or the device has to really become a part of that particular functional organ, or it has to basically refrain from attaching it to the blood. So, there are certain requirements which basically are very, very different for these applications, and that is basically leading us to understand more in detail that how a device can be supplied, how a device can be applied for restoring a certain functionality, and what sort of materials are required for engineering it. I will close my lecture here. Thank you.